Hi, welcome to Micro 205 Laboratory. So, to get started, what we're going to cover is some simple lab safety protocols. So, first thing, as always, you're going to be wearing your lab coat. This is going to protect you from anything splashing onto your skin. Gloves. So, this is focused on aseptic technique. You can contaminate your samples, but also you want to protect yourself still. So personal protective equipment such as gloves are important. Additionally, we also have lab safety goggles. Depending on the lab you work in, regulations could change. So for this lab, all you need is simple goggles that will cover your eyes, like so. So you have full protection above and mostly below for your eyes, such that nothing will get into your eyes. So sometimes when we're working, sometimes there might be aerosols, there might be something that splashes, you open a tube. You don't want to get any reagents such as crystal violet in your eyes or even alcohols or any other solutions that you don't know what they do. You also don't want to get any kind of microbes in your eyes as well. So that's why we want to wear our safety goggles. In some labs, you may also need and be required to wear a full face shield. And this again will help protect you against what you're working with. Another thing that we need to do while in the lab is wear closed-toed shoes. So the purpose of this is again safety. If we're wearing flip-flops or anything less than something that's covered, you can cut yourself. Again, you can drop things on you. You can step on things, and it's just again limiting your exposure and risk for contamination, for illness, or for injury. Okay, so something important when in lab is to make sure to always label your work. So things to start off with would be your initials, the sample or experiment you're doing. So here is, we're going to disinfect, and this is before, after, and then I'm going to be doing a keyboard. And then I'll do multiple keyboards as well just to see, but so this one will be from a computer that's just in the classroom, for example. So I'm going to put the date. So today is, we're going to do 10, 14, 18. And then if you're in the class, if you're in a class or whatever, you need to also put your class. So you could put MIC 205. We're here Monday, Wednesday, 8 a.m. So this is all the information that you want to have on your plate, for example, to make sure that people know that it's yours and that you know it's yours. So our initials, our sample right here, the date, our class section, and then also the time. So now, since I'm doing it before and after disinfectant, what I'm going to do is I'm going to take my plate, I'm going to draw a line down the middle of it. Okay? So on one side, I might put a B for before, then an A for after. So when I'm streaking, what I'm going to do is I'm going to take one of my sterile cotton swabs, and what I'm going to do is first wipe the keyboard. So I'm just going to wipe my keyboard. And then what I'm going to do is I'm going to streak to grow a lawn. So I'm going to do this. I'm going to take my swab and I'm going to streak on the entire surface such that every area is covered. So again, it's going to be just like this. And this, if you were doing aseptic technique, we would have a flame around the area we're working to help prevent contamination. So now what we're going to be looking at is our hand washing experiment. So the purpose of this experiment is to look at hypothesis testing and also to go over controlled variables and experimental variables. So let's think, what is a controlled variable? A controlled variable is something that is constant throughout the experiment. So for example, depending on where I swab my hand, that will be a controlled variable. So for my hand, when we do the hand washing experiment, I will swab between my index and my middle finger. And this is before and after I wash my hands. So what are experimental variables? Experimental variables are things that change. So you're trying to test a hypothesis. So if I have an experimental variable, that's going to be based on what my hypothesis typically is. So if I'm going to hypothesize that washing my hands with warm water versus cold water is better, so I might hypothesize, okay, I think that washing with hot water is better than cold water. So now in my group, one of us would wash our hands with hot water, one of us would wash our hands with cold water. Mind you, we'll be able to test an experimental value. However, since the hands are now different, that's then an experimental variable. So it's something that the controlled part would be we wipe between the fingers of our index and our middle finger. However, since it's two different samples, yourself and your lab partner, that is experimental. So it's not constant. So what I'm going to do is I'm going to remove my glove, and I'm going to do it in such a way that the inside of my other glove does not touch the inside of the glove I'm removing. So ways you can do this, you pull up on your glove, and then you basically hook through, and then I've pulled off one glove. So now what I'm going to do is take my sterile swab, and swab it between my fingers, like so. Okay? And then, after I do this, I'm going to plate onto sterile media. So, taking my already labeled plate, I'm now going to swipe and inoculate my auger with what I just collected on the swab from between my index and middle fingers. So I'm going to cover this up, and now I'm going to go wash my hands, and then afterwards I would re-streak. This was from an experiment that we did with hand washing. So, as we discussed, our hypothesis was that we would get less bacteria after washing our hands than before. So, take a moment to look at these and count out how many bacteria approximately we have before and after hand washing. So, please pause the video and take a moment to count the difference. Alright, so I hypothesized that washing my hands would result in less bacterial growth on one side after I wash my hands than before. So it looks like my hypothesis what may have been correct. However, one thing, this result is actually kind of interesting because we'll typically see more growth after washing our hands. So the reason being for this is because the process of washing our hands is going to actually release bacteria from the crevices in our skin. So that way, when you plate your washed hand, or where it's moist, you'll be able to pick up more bacteria. So what may have happened is, by the time I took my swab and wiped it against my hand again in the same location, is that maybe my hand was too dry for this effect to happen. So this is something that I would record in my lab book. So, was my hypothesis true or false? What results do I see, and what do my results mean? So, my discussion, my results, my results would be how many bacteria do I see before, how many do I see after, what number of different types of bacteria I see. So, if you look in the plate, typically things that you'll look for in your colonies are size. So, for example, like this guy here might be fungal, for example. You have a couple other different bacteria as well. So you'll look at size, you'll look at the shape of the colony, and then also look at the color of the colony. And this will give you some idea of how many bacteria that you have on your plate. So that is something that you'll have in your discussion, and then any conclusions that you could make. So from my hypothesis, from my hypothesis and my results, I could conclude that hand washing is important. Now, we know from lab, and we also know from practical exercise and lecture, that the reason why we wash our hands is to remove transient or temporary bacteria from our hands. So a study that was shown in hospitals in the early 20th century showed that when doctors washed their hands, there was a lower mortality rate in maternity wards. So the reason being is because doctors would be taking the bacteria from previous patients, and they would be taking that bacteria and then giving that bacteria to someone else. And that is what would be causing the infection. So other results that we have that are really interesting is we also did our disinfectant test. So disinfectant just to test to see the microbes around us. Okay, so if you remember, this was using alcohol wipes and swabbing a keyboard before and after alcohol wipes were used. So here we see difference in microbes. So before we have more bacteria, after we have less bacteria, but we also have fungal growth. So I'm going to flip this plate so that we can see some of that. So I'm keeping my plate closed, as should you if you were to do this experiment, because I don't know what exactly has grown up on this plate. So this again is a safety thing. So you can see fungal growth by filamentous growth on these guys here. And then here we see a lot of common bacteria, and a lot of these microbes, if you look at your hand washing plate, it makes sense for my keyboard to have similar microbes, similar bacterial colonies growing up as on my hands. Reason being is because my hands touch my keyboard a lot, so they're going to deposit some of the microbes that I have on my hands. So this is a good example of showing you how much and how many microbes are around us. So, you shouldn't do crazy things like go around licking surfaces or, wash your hands every time that you do something, because if you put your hand around your mouth, your eyes, you could just be infecting yourself.